Hello, my name is Olivia Sanderfoot, and I am a postdoctoral scholar at the University of California, Los Angeles. Today, I will be sharing some of the research I conducted as a graduate student at the University of Washington in Seattle. As part of my doctoral studies, I led a community science project to monitor birds in cities in the Pacific Northwest region of the United States during COVID-19 pandemic lockdowns. I later used data collected by our volunteers to investigate how habitat, weather, air pollution, and human mobility together influenced the distributions and detectability of birds in cities during this period of highly reduced human activity. I am thrilled to be here today to share the findings of our study with you as part of this symposium. As we all know, urbanization transforms the environment. As we move from more rural into more urban areas, we tend to see natural vegetation replaced by built structures, and this can lead to direct habitat loss for many wildlife species, including birds. However, the percent impervious surface is not the only environmental stressor that can influence birds. Along the urban to rural gradient, we tend to see an increase in light pollution, noise pollution, air pollution, and human disturbance, all of which are known to affect the health and behavior of birds and are important to consider when developing conservation plans. Yet, teasing out these specific impacts of these environmental stressors is often a challenge because they are closely correlated. For example, as we move from more rural into more urban areas, we might see an increase in both air pollution and noise pollution, which means that it's difficult to tease out the individual, collective, and interactive effects of those stressors on birds. COVID-19 pandemic lockdowns presented this unique opportunity to start to tease out some of these impacts because rapid declines in human activity altered the intensity of these urban stressors in some places. For example, some cities observed improvements in air quality, whereas others saw declines in noise pollution or artificial light at night. And this gave ecologists around the world this chance to start to think about how the impact of one urban stressor versus another influenced birds in their cities. Furthermore, ecologists have been able to leverage regional variation in the impacts of COVID-19 pandemic lockdowns to think more about how specific urban stressors influence birds. So some cities may have seen an increase in air pollution, whereas others saw a decrease in air pollution. Some saw improvements in noise pollution, whereas others did not. And this can be related to specific lockdown policies or compliance with those policies, or just how different lockdowns affected human activity in different areas. And that gives us this chance to compare and contrast avian responses across cities and link those to the underlying urban stressors driving those responses. I was interested in two overarching research questions. First, how does the intensity of urbanization and availability of bird feeders and bird baths influence avian habitat use? So if we think about the urban, suburban, rural matrix, where are birds spending time and how can different resources influence habitat use within this highly heterogeneous area? And how might that be different during a time of reduced human activity like we observed during COVID-19 pandemic lockdowns? Second, how do daily changes in weather, air pollution, and human mobility influence detection of birds? As I mentioned earlier, in the US Pacific Northwest, we didn't actually observe a decrease in air pollution. We observed declines in human mobility, but we didn't observe a corresponding improvement in air quality. So this gave us the opportunity to think about how background levels of weather and air pollution influenced birds during a period of suppressed human activity. To answer these questions, I launched a community science project known locally as the University of Washington Lockdown Birding Study. I invited volunteers to help us collect data to monitor birds in the US Pacific Northwest during the spring of 2020, which aligned with peak COVID-19 lockdowns in this region. We asked volunteers to conduct weekly 10-minute point counts and submit their observations as complete checklists to the eBird database. We asked volunteers to conduct point counts on a weekly basis, if possible, beginning April 1st and ending June 30th of that year. And we had a tremendous outpouring of support from the community for this project. More than 900 folks enrolled in the community science project. So we really did have this incredibly powerful, rich data set to work with for our analysis. Our final data set includes 6,640 eBird checklists, each of which represents a single survey or the results of one 10-minute point count. These surveys were conducted at 429 sites in Washington and Oregon, shown as dots in the map here. These surveys were conducted by a total of 376 unique observers. As you can see, our monitoring sites were heavily concentrated in major metropolitan areas in Washington and Oregon, such as Seattle and Portland, and that's why we ultimately decided to focus our analysis on observations collected in just these two states rather than across the US and Canadian Pacific Northwest. Together, our volunteers contributed hundreds of hours of time and observed 193 species. And we just want to say thank you once again for all of their help and support with this initiative. We could not have done this without them. In addition to the data collected by our volunteers, we had to pull in a number of covariates to conduct our analysis. We started with pulling data on land cover from the National Land Cover Database, and we pulled out two metrics. One, the land cover class, which we grouped into three categories: high developed, low developed, and natural areas. High developed included all areas with greater than 50% impervious cover, whereas low developed areas included those with less than 50% impervious cover. Natural areas included everything that was not considered developed. So in the context of our study, high developed would be busy, bustling downtown areas, whereas low developed would be more like a suburban backyard, and natural areas would likely be parks. Our volunteers did get to select their own monitoring sites, so we did not institute random spatial sampling, and most volunteers observed their backyards or a local green space. So all of these sites were within micropolitan and metropolitan areas within the urban to suburban matrix. We also pulled percent canopy cover from the National Land Cover Database as an additional metric to get at the intensity of urbanization. So between the land cover class and percent canopy cover, we feel like we have adequately characterized the intensity of urbanization across the sites included in our analysis. We also pulled data on weather from the North American Regional Reanalysis, including the daily mean air temperature and daily accumulated precipitation for each survey at each site. So for each survey, we assigned data on temperature and precipitation based on daily averages provided by this model. To characterize air pollution at monitoring sites, we relied on data from ground-based instruments included in the Environmental Protection Agency's air quality system. And we focused on characterizing the daily mean concentration of fine particulate matter at monitoring sites. Fine particulate matter refers to all suspended solid and liquid particles that are smaller than 2.5 microns in aerodynamic diameter. This is an air pollutant of increasing importance for public health and is known to be incredibly valuable in assessing the impact of urban air pollution on human populations, which is why we wanted to focus on this pollutant in our analysis. We also characterized human activity at monitoring sites using data available from the Google Community Mobility Reports. And this data set provides information on the percent change in human mobility based on cell phone locations compared to a pre-pandemic baseline. So for every day at every site, we were able to consider how the local change in human mobility may have influenced birds in that location. This is a plot showing the percent change in human mobility with the day of year at study sites included in our analysis. So you can see that from the beginning to end of our study period, human mobility ticked back up, but did not reach pre-pandemic levels, which are marked by the black line at y=0. So human mobility was suppressed across the study area for the duration of our study, but as lockdown restrictions eased between April and June, we did see folks slowly start to resume the activity that they exhibited prior to the lockdowns. In addition to this data, which was all available from public resources, we also invited our adult volunteers to participate in an online questionnaire to collect additional information, both about their monitoring sites and their motivations for volunteering. This questionnaire allowed us to collect data on site characteristics that we would never have been able to look at using data sets available to the public. We asked about whether or not seed or suet feeders were available to birds at monitoring sites, as well as whether or not hummingbirds were available and whether bird baths were available and how excited folks were about birding and what might allow them to participate in a project like this in the future. We received 284 responses to this questionnaire. So we did not have 100% participation in this component of our project, but this did allow us to conduct a secondary analysis looking at how some of these supplementary resources influenced avian habitat use. For our analysis, we focused on the 46 species most commonly observed by volunteers during spring of 2020, and we used single season occupancy models to look at the distribution and detection of those species across our study area. Occupancy models are commonly used by ecologists to explore bird distributions because they allow us to look at where birds occur on the landscape while accounting for imperfect detection, or instances where we fail to detect a bird even though it is present. This allows us to examine drivers of both occurrence and detection. So where are birds on the landscape and how likely are we to detect them at any given place and time? The definition of a site is extremely important in occupancy modeling because it relates directly to inference regarding which areas are occupied by a species. But it can be very challenging to determine if a site is occupied or not in continuous habitat. For example, in our study, the home ranges of our study species often exceeded the size of monitoring sites, which were 100 meters in diameter. And this means that species may not have been consistently present within a site or could have been present at more than one site. So we decided that we would interpret the probability of occupancy as the probability of use. That is the probability that a species visited a monitoring site at least once during the study period. So by including predictors of occupancy in our models, we were able to assess drivers of site use, or in other words, what attracted birds to specific locations within our study area. It's worth remembering that detectability reflects both the availability and perceptibility of birds, which means that it reflects both whether a bird could be detected as well as whether it is noted by an observer. So even if a species was known to use a site, it may not always have been available to be observed by volunteers at that location because it could have been elsewhere at the time the survey took place, it could have been hidden from view, or it could have been silent. So when we think about drivers of detection, we have to think both about how changes in behavior could have influenced the detectability of birds, as well as whether or not observers were able to pick up on visual and auditory cues of their presence. We considered a number of effects on site use, including land cover, percent canopy cover, bird feeders, hummingbird feeders, and bird baths. So again, we wanted to think about how the intensity of urbanization during this period of suppressed human activity influenced site use by birds. And to get at that, we considered both land cover class and percent canopy cover. In a secondary analysis, using data from that online questionnaire, we were able to think about how bird feeders, hummingbird feeders, and bird baths may have also attracted birds to monitoring sites. We also considered a number of fixed effects on detection. In the detection portion of our occupancy models, we included day of year and time of day, both as linear and quadratic effects. We also considered the effect of weekend on detection, as well as air temperature, accumulated precipitation, and PM 2.5. We knew that there would be seasonal variation in bird activity that influenced the probability of detecting birds, which is why we included day of year in our analysis. And we also knew that birds would exhibit day-to-day and daily changes in bird activity that ultimately influenced their detection, which is why we included time of day. We included weekend because we assumed that there would be differences in how people behaved during the weekdays and during the weekends that could influence detection of birds. Finally, we considered both weather and air pollution, specifically looking at air temperature, precipitation, and again, fine particulate matter, or PM 2.5. In addition to these covariates, we also considered how the percent change in human mobility influenced birds. So we wanted to account for seasonal variation, daily variation, differences in human activity on weekend, weather, and air pollution. And after all of that, consider how day-to-day changes in how people move around or the intensity of human disturbance influenced the detectability of birds. So we found that after accounting for seasonality, time of day, weather, and air quality, daily shifts in human mobility affected the detectability of more than 3/4 of our study species, which to us suggests that changes in human behavior during COVID-19 pandemic lockdowns in the Pacific Northwest influenced the availability and/or perceptibility of birds. We had expected that songbirds, which are often detected by ear, would be more easily detected when lockdown restrictions were more extreme at the beginning of our study period in April rather than in June, simply because as fewer people were moving about, there would likely be less background noise, which means that songbirds would be easier to hear. However, we didn't observe a consistent negative relationship between human mobility and detectability of songbirds, which suggested to us that our results can't be entirely explained by enhanced conditions for birding. There must be another explanation, and there must be some shift in the availability of birds and the perceptibility of birds that is driven by behavioral responses to reductions in human disturbance. So for example, for birds that exhibited this positive relationship between human mobility and detectability, we believe that those species may have been more active or more abundant at our monitoring sites as lockdown restrictions eased. So as lockdown restrictions eased and human mobility or human disturbance increased, perhaps those birds were more drawn to the places where our volunteers were monitoring birds during the spring of 2020, which tended to be backyards and local green spaces. Maybe those birds were just spending more time in places that were in general going to be more enticing to them as lockdown restrictions eased. Furthermore, we found that air pollution influenced detection of 22% of study species, which also suggests that air pollution, in addition to human mobility, is driving species-specific behavioral changes in birds. We also found that neither land cover nor canopy cover influenced site use by 50% of study species, which perhaps suggests that backyard birds may have been able to use a wider range of habitats during lockdowns. Land cover was also a more important driver than supplementary resources. So we found that while land cover or canopy cover influenced site use by 50% of study species, which was a smaller proportion than we expected, land cover was definitely still a more important driver than availability of bird feeders, bird baths, et cetera. So that shows us that when thinking about conservation plans for birds in urban areas, we really do need to think about habitat. Bird feeders and bird baths alone aren't going to draw birds into more highly developed areas. We did find that seed or suet feeders influenced site use by about 1/5 of our study species, but we definitely saw the strongest response to hummingbird feeders. Both hummingbird species included in our study were definitely drawn to hummingbird feeders. We also found that 13% of species were positively associated with bird baths, and no species exhibited a negative relationship with bird baths, which suggests that bird baths only serve to draw birds into places within the urban to suburban matrix, as opposed to seeing birds avoid those places. Our study provided a snapshot of the distributions and detectability of backyard birds in cities under lockdown in the US Pacific Northwest during COVID-19 lockdowns. And we're really excited to have contributed to this growing body of research on the effects of pandemic lockdowns on birds and other wildlife. We showed that changes in human behavior during lockdowns influenced the detectability of a wide range of species, which likely means that there's an interesting and complex relationship between human disturbance in urban areas and bird behavior that ultimately influences the probability of observing birds. Furthermore, we found that air pollution is an important component of the detection process. Air pollution influences whether or not we are able to observe birds, and that is fascinating because while we often consider weather when describing the detection and distributions of birds, we often fail to consider air pollution. So this study supports the notion that air pollution is an overlooked component of detectability. Our study also shows that there is a lot to be gained by conducting community science projects during times of reduced human activity to help us tease out the influence of urban stressors, as well as interactions with habitat and supplementary resources. And we really hope that we've inspired other teams to consider leveraging existing community science infrastructure to launch similar initiatives to support rapid response regional monitoring programs that can help us better understand how birds are responding to the individual, collective, and interactive effects of urban stressors along the urban to rural gradient. There were a number of bonus outcomes of this project that I want to quickly highlight. This generated community interest and support for our research, and the value of that can't be overestimated. And this project also allowed us to share a love of birds with beginner birders who were welcome and encouraged to participate in this project. We also felt like we were able to engage birders in community science and scientific inquiry, and give our community something positive to do during a difficult time. So I would definitely recommend it, and I'd be happy to answer questions about how we considered the caveats of engaging beginner birders and answering these questions using community science. Finally, I just want to acknowledge our funding. This project was funded by a National Science Foundation Graduate Research Fellowship Program, as well as the McIntyre Stennis Cooperative Forestry Research Program. I want to give a thank you to my co-authors, Drs. Joel Kaufman and Beth Gardner at the University of Washington, for all of their help with this analysis and the development of our manuscript. And once more, I have to give a shout out to our amazing volunteers, without which this work would have been impossible. So with that, I'd be happy to take any questions in the chat. And if we don't have time to get to your specific questions at the symposium today, you are welcome to learn more about my work at my website or send me an email at osanderhood@ucla.edu. And I'd be happy to set up a time to chat more about our work and your work and opportunities to collaborate and learn more about how urban stressors changed in cities around the world and what that meant for the birds that we all love. Thank you so much for your time and for your participation in this symposium. I'm so excited to be here, and I can't wait for the next event. Thanks so much and have a great day.